Two pioneer microbiologists laid the foundation for studying the role of microbes in natural sulfur cycling, representing two halves of the cycle. The Russian microbiologist Sergei Vinogradsky discovered aerobic filamentous sulfur-oxidizing chemolithoautotrophic bacteria, and the founder of the Dutch School of Microbiology, Martinus Beyering, described first anaerobic sulfate-respiring bacterium, now known as the sulfovibrio. I am a Russian microbiologist from the Vinogradsky Institute in Moscow, but I also working in parallel in TU Delft, particularly on sulfur microbiology. In this lecture, I will briefly introduce the basic knowledge about sulfur cycling microbes, including their ecology, metabolic features, and enzymology. Sulfur is the first, most abundant element on Earth, and it is very important for both geochemistry and biogeochemistry. It exists in a large variety of natural compounds, with sulfur valency from -2 to +6, thus participating in redox reactions as both electron donor and acceptor, resulting in circular transformation of sulfur compounds from most reduced state to most oxidized. There are three more stable inorganic sulfur compounds, which are also dominated in total amounts: metal sulfides with sulfur valency -2, elemental sulfur valency 0, and sulfate valency +6. Compounds with intermediate or mixed sulfur valency, such as thiosulfate or polythionates, are also part of the cycle, but they are transient due to their chemical reactivity. Sulfur is linked with other global element cycles, including nitrogen and carbon cycles, serving either as the electron donor for CO2 fixation or denitrification, as the electron acceptor for anaerobic organic carbon oxidation. Another important interaction is with heavy metals, forming insoluble metal sulfides. The sulfur-based redox transformations can go spontaneously without microbes, but there are two very important factors that make enzymatic microbial catalysis far superior. First is the microbial enzymes, as a rule, have 2-3 orders of magnitude higher substrate affinity in micromolar rather than in millimolar range. Two, microbial catalysis has high rates at normal conditions, while chemical catalysis often need much higher temperature and pressure. The microbially mediated redox sulfur cycle has two branches. The oxidative branch starts with sulfide and goes through intermediate 2-electron oxidation to 0-valent sulfur, which is very important for application. Next, sulfur is oxidized all the way to sulfate. Thiosulfate is formed by chemical oxidation of sulfide and then is oxidized further to sulfate by microbes. Such microbes are called sulfur oxidizing. They can be either aerobic or anaerobic, for example, phototrophic or denitrifying. The opposite reductive branch of sulfur cycle starts with either elemental sulfur, it's called sulfur respiration, or sulfate, it's called dissimilatory sulfate reduction, and results in sulfide as final product. There is also a hybrid anaerobic pathway whereby the same sulfur compound with an intermediate valency can serve as both electron donor and electron acceptor, such as sulfur, thiosulfate, and sulfide, resulting in formation of both fully reduced and fully oxidized products. Such reactions are called disproportionation. Here we start with the oxidative branch, which is represented by sulfur-oxidizing bacteria and archaea. The most important and efficient type of such microorganisms is the group of chemolithoautotrophs, which means that they are using reduced inorganic compounds as the energy source and electron donors for assimilation of inorganic compounds, either CO2 or bicarbonate, into biomass. The range of sulfur compounds utilized by such organisms is relatively broad, with a maximum of 8 electrons extracted from the sulfur atom, which yield as much energy in the form of ATP and membrane potential as, for example, aerobic oxidation of acetate to CO2. However, since such microbes must invest a significant portion of their energy to produce NADH for CO2 reduction, their biomass yield is much lower than in heterotrophs. The most common place where sulfur-oxidizing microbes can be found is the redox zone in stratified aquatic habitats, whereby two opposed gradients of sulfide coming from the anaerobic sediments and dissolved oxygen coming from the surface oxic waters are overlapping in a narrow layer at micromolar concentrations, too low for chemical oxidation of sulfide, but optimal for sulfur oxidizers. There, they form dense biomass acting as sulfide filter. Such a system can be mimicked in laboratory using a simple technique known as the sulfide oxygen gradient culture and can be used to enrich and isolate sulfide utilizing bacteria in PU culture. The most common reaction employed by sulfur-oxidizing microbes include complete oxidation of sulfide and thiosulfate to sulfate with 8 electrons, used mostly at neutral and alkaline pH, since H2S is present in its dissociated form as hydrosulfide, which is soluble and less toxic for cells, and thiosulfate is chemically stable. Polythionates and elemental sulfur are stable at moderately and highly acidic conditions, respectively, and are used by acidophilic sulfur-oxidizing bacteria and archaea. Thiocyanate is a unique inorganic sulfur compound with the C-S bond and a very stable nitrile bond between C and nitrogen. Microbial-based oxidation of thiocyanate to sulfate is an important industrial process. Finally, a few sulfur-oxidizing chemolithotropes can grow anaerobically with sulfide or thiosulfate using nitrogen oxides as acceptor instead of oxygen. This process is known as thiodenitrification. Sulfide can be oxidized to zero-valent sulfur by two periplasmic sulfide dehydrogenases, one using quinone and another cytochrome C as the electron acceptors. This reaction is usually fast. Further sulfur oxidation to sulfate is a much slower process, which involved a cascade of sulfur transfer stages into a cytoplasm, whereby two different enzymatic systems can perform the 6-electron oxidation of elemental sulfur. The most well-studied enzymatic system is the periplasmic SOX cycle, present in a very broad range of sulfur-oxidizing bacteria for thiosulfate oxidation to sulfate. It includes several enzyme complexes performing 3 types of sulfur atom transformations: sulfur atom chelating and transferring from one complex to another, sulfur atom oxidation, and cleavage of oxidized sulfur with the release of sulfate.